Sí, sí. Sí, bueno, audio uno, 2, 3, sí. Bueno, 2, 2, 2.
Hola, buen día a todos. Gracias por venir. Okay. Good morning and welcome. On behalf of the Centro de Investigaciones Regionales, Dr. Hideo Noguchi, and the Department of Neuroscience, I'm pleased to welcome you to today's lectures that the name is Pathway to Autism, How Genetic Variants Disrupt Neuronal Cell Biology. For me, it's a grand pleasure present and introduce our speaker, Dr. Christopher C. Quinn, yeah, that is associate professor in the Department of Biological Science at the University of Wisconsin at Milwaukee. And actually, it's a co-director of the neuroscience program in this university. Dr. Quinn earned his Ph.D. in neurobiology from Yale University. He completed his postdoctoral training at Rutgers University before joining the faculty at the University of Wisconsin in Milwaukee in 2010. Since then, he has built a distinguished career on research and teaching in the development biology, genetics, and cell biology. And right now, Dr. Quinn's research explores the cellular and molecular mechanisms behind neurodevelopmental and neurodegenerative disease. His lab used model organisms such as C. elegans to study how to autism-related genetic variants affect neuronal development, organelle function, and axonal health. His work has revealed how changes in genes such as calcium channels and ribosomal proteins can alter endosomal dynamics, mitochondrial functions, and neuronal connections, thereby leading to neurological problems. He has published many articles in top science journals, including Nature Cell Biology, PLOS ONE, PLOS Biology, Journal of Neuroscience, among others. He has also received several honors, including the Shaw Scientist Award and the Individual National Research Service Award. In today's lectures, Dr. Quinn will explain how new genetic variants likened to autism and intellectual disability can disrupt key parts of the neuronal cell biology, helping us to understand the connection between human genetics and cellular problems. Please join me in welcome to Dr. Christopher C. Quinn. The auditor is yours. Thanks a lot for coming.
All right, thank you. All right, can everyone hear me? All right, is this, yeah, that's working now. All right, great, thank you. So yeah, it's a real pleasure to be here at the Autonomous University of the Yucatan. And I'd like to start by thanking Dr. Humberto for all his hard work in helping to organize my visit here today. It's also really great to see so many students in the audience as well. So I'm going to tell you about some of the work from my laboratory, and all this work is done using a model organism known as C. elegans. Right? So this is actually a worm, but it's a microscopic worm, so it's very, very small. You can only see it using a microscope. And the reason we use it is because it's very simple. So it has just about 900 cells and only about 300 neurons, right? So there are 300 neurons, but the work I'm going to tell you about today actually just focuses on one of these neurons. It's the PLM neuron. So this neuron has the cell body in the tail of the worm, and then it extends its axon laterally along the body wall, just like this, and then the axon stops right in the mid-body. right? So this is the PLM axon. And what we're interested in is trying to figure out how does this axon know where to grow, and specifically, how does it know where to stop and form neuronal connections? Okay, so although we do all of our experiments using this microscopic worm, what we're really interested in is the human brain, right? And we're interested in how axons actually form connections within the human brain. And why we're interested in this is because when things go wrong with this process, that creates what we call neurodevelopmental disorders and neuropsychiatric disorders. And one example, and a lot of our focus is on autism, right? So you can see an example here on the left side, right here, you can see this is what's known as the corpus callosum. So that's a major fiber track or axonal track in the human brain. It connects the left side of the brain with the right side of the brain. And you can see this is very nicely organized. This is a child with autism. And you can see that the corpus callosum is disorganized and it's not developed as well. So we think that these problems with the axonal trajectories or axonal connections are what are causing neuropsychiatric and neurodevelopmental disorders. So by the way, if there are any questions as I go, I'm more than happy, if you just raise your hand, more than happy to answer questions during the course of the talk. Yeah, so here's another example. This is just one other example. So this is another axonal tract that's known as the mesolimbic pathway. And within the human brain, this mesolimbic pathway is thought to mediate reward. So if you do something good happens to you, the mesolimbic pathway makes you feel good. Now this is normal, shown in red. These are the axons in a normal child. And you can see these are the axonal pathways in a child with autism. So you can see this is underdeveloped by quite a bit. So again, we think that these defects in the axonal connections are really what's underlying the disorder of autism. Yeah. Yeah, I'm sorry, I actually don't know the answer to your question. Yeah, because mostly because I don't study like the human nervous system. Yeah, but a lot of it is certainly dopaminergic, I'm sure. I'm just not sure if it's exclusively dopaminergic.
Because if it's effective in child with autism, maybe they don't experience pleasure as we do.
Yeah.
But I think
that's the idea. Yeah, that's the idea.
I ask you because I work with the
Yeah. Yeah, no, I think it's definitely worth looking into that some more to try to understand that. But yeah, that's the basic idea is if a child has autism, they're not responding to the stimuli. Like it may not be like a social interaction might not be as rewarding, right? Or normal types of things. And that's maybe a big part of what's affecting their behavior. And I should say that these are just two examples. There's actually many examples of axonal pathways that are disrupted in autism. So it's a pretty active field. It's been going on a while, as many times not clear, but at any rate, this is just some examples of how axons are affected. So that's how the axons are affected. But my real interest and my approach is with genetics, right? And it turns out, so there's been a lot of work in understanding human genetics of autism in probably the past 10 years. And a lot of this has to do with whole genome sequencing, right? So this is something that's been getting started in the past 10 or so years. And the way this works, so that the goal then is to identify individual genes that are associated with autism. So the idea is that mutations or variants in these genes could be causing the axonal defects, by the way, the axonal defects are just one aspect. There's many others, like there's a lot of problems with synapses, for instance, right? But it's just for my work, I'm just focusing on the axons, but there are many different problems in the brain. But anyway, the way this works is that the geneticists or the investigators will obtain genome sequencing from a family. And this family is always that they choose will always have a dad and a mom. And the dad and the mom will be normal, like they don't have any autism or any disease. And then they also obtain a sequence of the child, and this child is going to have autism, right? So they take the sequence from these 3 individuals, and then they search for mutations that are in the child, but not in the father or the mother. So these are not familial, they're not passed down through the family, they're brand new mutations that are arising just in the child. And where they come from, usually it's from the dad's sperm, right? So the dad has the genome is normal, but there's a brand new mutation, which we call de novo, that's appearing just in the sperm, right? So that's the idea. And then what happens is they don't do just one family, they get sequenced from thousands of families, and they start to search for genes that seem to be accumulating these mutations and at a population level at a rate that's greater than expected by chance. And then it's a lot of statistical and mathematical analysis. And right now there's about 200 genes, but depends on the statistical cutoffs, but there's between 200 to 500 genes that have been associated with autism in this way, right? And this is just, we're just an example, like each circle here represents one of those genes, and it's kind of showing you how many mutations have been identified in those genes. So the goal of my laboratory then is to take those genes that have been identified by human geneticists, right? So there's other scientists that are working on human genetics. They identify the genes, and then we take those genes and those mutations and try to figure out how they're affecting the axon development. So it's the development of the nervous system. And we do that using C. elegans, as I explained earlier. So I'm going to talk about 2 different projects today, if we have time, but I'm going to start with this one. It's called CACNA1C. So this is a calcium channel, and this calcium channel actually has been implicated a lot in autism, and actually all neuropsychiatric disorders, this calcium channel seems to be involved. So I'm going to tell you how we use C. elegans to study this, how do mutations in this channel cause defects in axon development. The other part, if we have time, I'll get into a different project. This is an RNA binding protein. It's called RBM27. And this protein, this gene also has been identified in children with autism. And this is one that binds, this is a protein that binds to RNA. And we found that these mutations are causing degeneration of axons in young worms. All right, so first thing I want to talk about the calcium channel. So many years ago, actually back in 2013, so it's a long time ago, there was a paper published where they were searching for genes that are associated with multiple neuropsychiatric disorders. So the disorders they were thinking about or looking for were autism. This one is ADHD, bipolar disorder, schizophrenia, and major depression. So it's 5 different neuropsychiatric disorders. And they were using this type of genetic analysis to try to identify genes that are associated with all 5. So something that's really influenced in neuropsychiatric disorders. And they actually only found 4 different genes with association. So, but what really caught my attention is that out of these 4 genes, these are the ones circled in red, 2 of them actually encode proteins that are part of the same calcium channel, right? So this is a calcium channel, and this is what's known as a voltage-gated calcium channel. So what that means, it's been studied a lot in neural transmission. What that means, voltage-gated calcium channel, it's there's a voltage, and the voltage causes the channel to open, and then calcium flows through the channel. And these mutations affect a protein called CACNA1C, which is actually, that's the pore-forming subunit. So calcium flows through this channel. And then there's this other thing, the other protein is called CACN-B2. And this is what we call an accessory subunit. So it helps control the channel. But the other very interesting thing is that there have been these very, very rare. So by the way, these mutations identified here in this study, these are called common mutations or common variants. So they have actually a very small effect on the proteins, and they're very widespread in the human population. So for example, these mutations that were identified, they're present in about 1/3 of the human population, but they have a very, very small effect. So it does not cause trouble in most people, and they're just considered risk factors, right? So very, very small effect. But there's these other very rare mutations in the same gene, right? So this is also in the CACNA1C gene. And these are really the de novo variants. So they're very, very rare, and they have a very strong effect. So just having this one mutation causes this really terrible disease known as Timothy syndrome, right? And why Timothy syndrome is so terrible? Because the heart, it affects the heart, and the heart doesn't work right. And most of these kids will die. Almost all of them die before becoming adults. So they'll all die as children, and it's all because of the heart problems. And the reason is because calcium is very important for regulating the heart function. But there's another phenotype that I'm interested in, which is autism, right? And intellectual disability. So all these kids also have autism, and it's very severe. So they have severe cases of autism. And you can see here, there's just other things like there's problems with their hands and feet and also their faces. But again, the worst part is the heart. But my focus is completely on autism, though. So these are all gain-of-function mutations. So what it means is that the channel is overactive. It's letting too much calcium into the cell, right, or into the organelle. It's just passing too much calcium through. So what we did, actually, when we first started studying this, there had been a lot of labs that had studied Timothy syndrome mutation in the heart, because that's the worst part of the disease. But we know there's also this autism, so it must be affecting the brain. We want to know, nobody had looked at how this affected the brain. So we wanted to use C. elegans as our model organism to address this question. So what we did then is we use genome editing. So this is CRISPR, and we inserted the same mutation. So you can see here, right here is the amino acid sequence from humans. And this is the mutation. So normally this should be a glycine, and it changes into an arginine. And that one amino acid change is what's causing so much calcium, like too much calcium to flow through the channel. So we want to create a model in C. elegans. So the way we do that is using CRISPR, and we can, we use CRISPR to edit the genome, and we can take the same mutation from human and we can put it into the worm, right? Because the worm genes are different than human genes, but there's a lot of similarity. There's many amino acids, many parts of the protein are very similar. And you can see in this part of the protein, it's almost exactly the same between humans and worms. So this is an alignment, it's the same amino acids in worms as you see in humans, mostly. You just see very few differences. So we can use this as a good model to, we can use the worms to understand what's going wrong in humans. So we did that. We used our CRISPR or our genome editing. And we put this mutation into the worms, and then we look at the axons and we ask, what's gone wrong here? And let me go back to the beginning. So I want you to show you a little bit more. So because the figures I'm going to show you now are sort of like just focused on one area, but here's kind of the view of the whole worm. So as I said, this PLM has its the cell body in the tail, and extends its axon, and then it stops right here in the mid body. And it's actually very important that it stops there, because this is a sensor for light touch. And this other neuron here controls it's a sensor of touch in the anterior portion, in the posterior portion. So anyway, the point is this axon should stop right here in the middle of the worm. So what we found though, in our experiment, what we found in our mutant, so this is focused right on the middle, and you can see this axon, it stops right here where it should. But in our mutant, the axon grows too far, right? So it's invading, essentially it's invading the territory of this other neuron. So that's, that's not good, and we think that can affect the behavior of the worm. So that, that's the, that's the idea here. So we took, so again, just to summarize that, we took the mutation from humans, from children that have autism, this Timothy syndrome. We used genome editing to put that into the worm. And then what we found is when we did that, we get this defect in the axon, right? So it's growing too far. That's the idea. All right. So are there any questions about that? Because that's sort of the most important thing I want to tell you about. Yeah. Any questions? Yeah. Yeah, so that's a very good question, and I get that question very often. So that's the only, okay, so we've only looked at 2 axons, actually this one, and it goes too far, but this axon here is also affected, we know that. So we have actually a separate publication that showed that, and this axon also goes too far. So normally that one, it goes into the head, and it should stop before reaching the nose, but it goes all the way into the nose. So yeah, this other one is also affected. Now, probably there's other, there's 300 neurons. I bet other ones are affected, but we've only looked at these 2, and both of them are affected. But yeah, thank you. Yeah.
So that calcium channel voltage, gated calcium channel, belongs to one of the families that are usually at the end of the synaptic button.
Yeah, exactly. Yeah, so this is yeah, so you're challenging me because I'm not into physiology too much, but I think it's a Q type. But yeah, it is one of those that's involved. It's very extensively studied. Exactly. So yeah, usually what happens is that there's a voltage arises at the synaptic button, and it allows it activates this channel, allows calcium to flow in, and that triggers release of neurotransmitters. That's the very well studied and understood aspect of this. What I'm telling you now is something that's, very new information that just came out of my laboratory, that it's also affecting development, and it's a different cause, right? This is not probably anything to do with neurotransmission. So it's sort of a newer function for this channel. But that's a very good question. Thank you. So yeah, anyway, that's that. So we did find, though, we looked a little bit at behavior. So we mostly just study axons, but some other people have encouraged us to look a little at behavior. And we did find these mutants, there is a change in their behavior. So this this blue line represents the wild type. These are normal worms. And what we do is we touch the worm and see how often does it respond to our touch. And we see what's known as habituation. So each time you touch the worm, normally it responds less and less. So it's habituating. And you see that here, there's a graph. So first time, 80% of them respond. Second time, most of them respond. But after that, it's less and less. So in our mutants, the same thing happens, but it happens much more rapidly. So we'll see first time, most of them respond, and then there's a big drop off in the response rate. So we do think, we haven't studied behavior too much, but we do think that this mutation, aside from affecting the axons, is also affecting the behavior. So again, getting back to Jose's question, again, so I mentioned we think that this is not working, through neurotransmission. It's doing something else. And that's what we're really trying to work on right now. Like, how is this channel disrupting cell biology to cause these axonal defects? And this is like ongoing work. We don't really know the answers yet. But one thing we did find is that it appears So we use a green fluorescent protein. So how many, can you just raise your hand if you're familiar with this, like green fluorescent protein or protein labeling? Yeah, so what it is, yeah, so thank you. So it's a fluorescent protein. It came from jellyfish and it glows green, right? So we can use that to visualize the localization of proteins, like where's a protein localized. So we want to know where is this calcium channel localized in the neuron. And we already know that it's localized in the synapse. That's been studied a lot, but we want to know where else is it localized, because that could give us a clue on what it's doing. And what we found out, so here it is, this is the green EGO19. So EGO19, by the way, that's the name of the calcium channel in worms. We call it EGO19. We saw that it was localized in these spots. So this is along the axon and it's these spots. So these are not synapses. There's no synapses at those locations, but we did see these spots. And what we did then, when we saw them, we were like, okay, that looks, we didn't know. We were just guessing, like, what could these spots be? And we tested a few different things, but we thought it was some type of vesicle, and we tested markers for different vesicles. And what we found out is that it's co-localizing with late endosomes. So wherever you see a green spot, so this is actually the same worm. We just took the picture twice, once with the green color, once with the red color. And we found that this channel was always localized with these vesicles that are late endosomes. So this is a late endosome. It's just a vesicle within, it's a vesicle, right? And it moves things around. So these vesicles, by the way, this is a still picture, and I don't have any movies to show you, but if I had a movie, you would see these vesicles moving all around, right? They're not still, they move all over. So that was kind of a clue. So we think, this doesn't prove anything, but it gave us the idea that this mutation might be affecting the function of these vesicles, these late endosomes. Yeah, no problem. Yeah.
First question, do these endosomes are like endocytic vesicles taken by the cells at certain parts of the membrane? Is that an endosome?
Okay, so that is an endosome. There's different kinds of endosomes. What you're talking about is the early endosome, right? And that receives things by endocytosis directly. These are just like further down the pathway. So there's early endosomes, and then there's late endosomes, and then there's lysosomes. And it's like, yeah, it starts with early endosome, but then, yeah, that would be passed through to a late endosome, and then that would eventually go to a lysosome. Yeah, I think so. I think that's probably where it originates.
Do these late endosomes participate in axonal transport?
Yes.
That is from the synaptic button to the soma and vice versa. So the soma.
Yeah, that's.
Trafficking.
Yeah, exactly.
Because I'm thinking that maybe the overfunction of this calcium channel at the end of the axon is promoting that these endosomes appear.
Yeah.
So they're transmitting something to the cell nucleus.
Yeah.
Because how to explain that the axon is growing a longer distance, but the only explanation is that the nucleus is exporting something so the axon can grow.
Yeah,
But
that's.
I'm thinking of that.
Yeah.
I'm not sure if I am okay.
No, we're thinking along. I mean, we don't have the evidence for that yet, but that's what we're trying to investigate. Is that, yeah, these mutations are affecting neuronal transport, right? And we think, so we actually, it's still hypothetical, but what I'm proposing is that it's affecting transport of certain receptors to the tip of the axon, and those receptors are controlling growth of the axon. That's the idea, but we don't know that for sure, but we're working on that. We're trying to do experiments. But thank you, very good question, right on the point. And so here, what we did is we looked at our mutants. So again, I don't have movies, but as I said, these are the endosomes and they are moving. So if you looked at this live or in a movie, you see them moving all along. But we looked at the motility, and we find normally about 60% of them are moving, The other 40% are sitting still. That's in wild type or the normal worms. But in our mutants, so these are all just two different alleles, two different versions of the same mutation. We find only about 30% of them are moving. So clearly, as you got to it, this mutation is affecting motility of these late endosomes, right? So it's definitely should be affecting transport of something. Again, we don't know what, but I'm hypothesizing that it's specific receptors that are driving axon growth. That's something, yeah, that's what I'm working on, trying to get a new source of funding to fund those studies, but something we want to work on more in the future. Thank you. Okay, so yeah, it's certainly affecting that. And then, as I said, we are looking at how it affects receptors, but there's another thing that we think that it does affect, and we have some more evidence for this. This one is called, in C. elegans, we call it SHIN1. Now in humans, it's called SHANK, right? It's a SHANK protein. Now why I mention this is because SHANK is another gene, another protein that's also associated with autism, and it's been studied a lot. So there's many children that have autism and they have mutations in this SHANK gene. So we actually, what we did is we didn't want to figure out, what is being transported, what is being affected by our calcium channel. So we did a screen. So we screened, there's actually 100, we screened through like 100 different autism-associated genes to see which ones might be interacting. And the one that we came up with is the SHIN1 or SHANK, right? So we specifically, we were just trying a bunch of different autism-related proteins, and this is what we found out. But anyway, what we did find is that normally this SHANK protein, so this is not a receptor, it's intracellular, it's inside the neuron. It's concentrated right at the tip of this axon, right? And I have other data, I'm not going to show you today, but I have genetic data that tells us that this SHANK protein is actually helping the axon to stop growing, right? So when we have mutations, the axon grows too far. It's actually the same phenotype that we see with the mutation of the calcium channel. But the other thing we found though, so this is the localization. Again, this is labeled with GFP. Now in this is the calcium channel mutant, and you'll see you can still see a little bit of that SHANK at the tip, but there's a lot less, right? So we think that our calcium channel is interfering with transport of this SHANK to the tip of the axon. We don't know if that's direct or indirect, but apparently it's definitely doing that. And we think that's a major cause for the phenotype. And again, it all goes back to neuronal transport. And then this is just, we won't go through the graphs, but this is just the quantitative data. So when we do these experiments, we can't just analyze, we analyze at least like 40, I think in this experiment, we looked at 80 axons. So it's 80, I'm just showing you one example here, but we analyze by 80, and then each of those is a dot on the graph. And then the averages are shown with the bar. But anyway, this is wild type, so you can see, and we're just measuring how bright, what is the intensity of the protein at the tip. And you can see it's, I don't know, around 750 or so in the wild type. Then it's about 250 in the calcium channel mutant. So the idea, this is just a quantification of what you see here. But the point is that in our mutants, there's a lot less of this SHANK protein. And we find that really interesting because we know that the SHANK protein is also involved with autism. Okay, so, one other thing we did. So we want to know, I told you that having less, our mutant causes less SHANK protein. But the other question is, does this actually have anything to do with the phenotype, right? Or is it just a coincidence? And the way we can address that is we can replace that SHANK protein. So we use something called a transgene, and we can artificially increase expression of SHANK. And then what we find is that phenotype goes away. So we're essentially curing, or at least partially preventing that phenotype. So the way this works, in our EGL19 mutant, we see like about 48%, so about half of the axons are extending too far. But when we overexpress the shank protein, so we're putting more shank, that's reduced to 29%. So it's not completely cured, but it's partially, what we say, a partial suppression. And so that really gives us an idea that, yes, this deficit or this reduction in shank is responsible for that phenotype. Okay, so now I can get, that was the first part of my talk, so I'm gonna move through. Are there any other questions on the first part? Yeah. Yeah, it prevents the location of that shank at the tip of the axon. And why that's interesting is because, although I didn't show you, we have other data telling us that this shank protein tells the axon when to stop growing, right? So the idea is the calcium channel prevents localization of this protein to the axon tip. And that's why the axon doesn't stop growing, because it's missing, kind of the signal to stop. That's the idea there. And again, this all ties back to transport, axonal transport. Yes, so this, not only has it been found, but it's been found to be a cause for autism, right? So if you have a mutation in shank, it's a cause for autism, right? As is the EGL19, the calcium channel. So that's why we're so interested in this. And we're trying to figure out how these different things work together, right? Because right now, from human genetics, we have a big list, at least 100 genes that are tied to autism. But we really want to know, how do these interact? How do they work together to cause a disease? Like, we don't want to know just, and those are things like, so people are studying humans, like human genetics, that's all they can do is produce that list. They can't study how they work together. And that's why we're using worms, right? That's the idea here. Yeah. Yeah.
Increase the probability of the calcium in the cell and produce an instability, or this calcium and their microdome activated, for example, calcium modified for another proteins that go to phosphorylate, for example, or another genes, and produce these mechanisms going on the axon.
Yeah. So this mutation has actually been studied a lot through neurophysiology, and what they found is that it's open. It's letting too much calcium into the cell, and what happens is that there's a mutation in a part of the channel that causes deactivation, right? So basically, the calcium channel is not deactivating as well as it should, because normally, it can activate and deactivate, but it's staying open too long, letting too much calcium in. And what we think, so I haven't mentioned this too much, but we're working on this as hypothetical, but we think that it's actually controlling release of calcium from those late endosomes, and that's what's affecting their motility, right? That's the idea. Because we don't think this has to do with neurotransmission or the synapse, but we think that this channel is located on late endosomes. And other people, it's been shown by others that calcium can affect the movements of late endosomes. So that's our thinking. Thank you. All right, anything else before I move on? Yeah. Oh, sorry.
Could it be that these changes in axon development could also be related to strengths or some benefits that we have seen on autism people? I mean, what evolution has showed us is that often the changes and mutations come with a deal or a trade-off.
Uh-huh.
So we are sure that autism persons have intense hyperfocus, superior pattern recognition, and logical reasoning.
Yeah.
So even though these neurons are disorganized, but they work a little different of the neurotypical people. So could it be that these changes are also related with these strengths?
Yeah, I mean, absolutely, it could be. And because there's probably many different axonal tracts in the brain that are affected. I just showed you like 2 examples. There's many others. And absolutely, like, some of this, yeah, there's disorganization. There could also be more connectivity in certain areas. And in fact, I think, I focused more on things that look like they were wrong or deficits, but I do know that there is other work on axons in humans that show enhanced connectivity in certain aspects. So you're definitely right. That's an important area of autism research as well. Thank you. Okay.
I'm intrigued about the certificate chloroquine, which is a drug used for malaria, if I'm not mistaken.
Oh, chloroquine, yes.
But do chloroquine blocks this channel?
So the reason we use chloroquine, not because it affects calcium, it's because it affects the endosomal system. All right. So what chloroquine does, I think in terms of malaria, it prevents endocytosis, I think. Yeah, because the malaria gets into the neurons or into cells through endocytosis, right? And it turns out that chloroquine also affects the late endosomes. And I didn't show you, but we have another publication in worms where we treat them with chloroquine and we find that it definitely disturbs their late endosomes, right? So it affects the late endosomes. So that's why we use chloroquine, because our idea is that the EGL19 channel is affecting calcium and that's affecting motility of late endosomes. So we just wanted to find a different way to disrupt late endosomes. And that's what we did. So we use chloroquine. Yeah.
Apparently, it shortens the axon wild type.
Yeah. So what it does, actually, this is actually the intensity of the shank protein at the axon tip. So there's less shank at the tip. So that's the same thing that we see with our EGL19, our calcium channel gain of function. We see the same phenotype. And we should test that. We haven't done it yet, but we're going to do it to see how chloroquine affects the growth of the axon. Still something we need to do. But for now, we know that it affects the localization at the tip. So really our reason for doing this is just to try a different approach, right? We did one thing with genetics. We'll do something else with the drug chloroquine. But yeah, it's something that's come in handy. It's been useful for us. Thank you. All right, anything else before I move on? Yeah. All right, so I'll go. So we just have a little bit of time left, so I'll just try to give a kind of a very brief overview of this. So this is a different gene, right? But it's also been named as a candidate gene for autism. And the reason is because there's been 5 different mutations found within children that have autism in this gene. And this is called RBM27. Yeah, RBM27 in humans. In worms, we call it RBM26, but in humans, it's RBM27. Anyway, it's been, and actually this gene, by the way, has been studied very little. So my lab has 1 paper on it in worms, and then in the humans, there's only 1 or 2 papers published on this. So it's very, very little study. And part of the reason it's been studied so little is because although these mutations have been found there, these are all missense mutations. They're just changing one amino acid to another. So we actually don't know, or it was not known, does this change even affect the protein? Because many people, there's many such mutations that have no effect. You can change just one amino acid to another amino acid. Protein is fine, right? So because of this, it's gotten very little attention, even though it was named strong. So it's named strong candidate, but we don't know if it's really a good candidate. So we actually wanted to address this question using worms. And what we did then is so there's actually 5 mutations found in humans, but only 2 of the mutations actually have a similar amino acid in worms. So we focused on those 2. And again, we use the genome editing. So we also call this CRISPR. And same thing I told you about before. We take these mutations that were found in children, and we're going to put them into the worm. And when we did that, first thing we did was just look at protein expression, right? So we just looked at the protein expression, and this is the normal worms, and these dark lines here, that's our Western blot. So on a gel, then we can see expression of this RBM26 protein. And you can see there's a lot, like 2 dark bands, some light bands in between. Now here's one of the mutations. So again, this is a mutation that came out of children with autism. And you can see there's very little protein. And same with this other mutation, there's more, but still very little. So what this is telling us, although it's just changing one amino acid to another, a very small difference in the protein is probably becoming very unstable, right? And there's very little of it there. So because of this, we now know that these mutations are very important. They cause a big drop, you're essentially eradicating most of the protein. But we also wanted to see, how does this affect the phenotype? And now what I'm going to tell you now is a different kind of phenotype than what before I talked about the axon growing too far. Well, here, what we found, so I'm going to focus, we did find the axon grow too far, but our main phenotype we focused on was what we call axon beating. So we think this is a degeneration phenotype. So normally the axon should be nice and straight, but here it's getting these swollen spots. And these are something similar has been observed in human, like neurodegenerative diseases, right? There's cases where they've seen these swollen axons, like in postmortem brain of people that had, like Parkinson's disease, for instance. So yeah, that's the phenotype that we found. So I'm going to skip through this, maybe not as important, but just the overview is we wanted to know, does this gene work in the neuron or in the surrounding cells? And what we found, we did many different experiments, and we found very good evidence that it's actually working inside the neuron. So the idea is this protein works inside the neuron, and it's protecting the neuron from degeneration. So we will skip through that. And the other thing we found is that it's affecting the mitochondria, right? So mitochondria are already known to be very important in neuron degeneration, very important for Parkinson's and Alzheimer's. And that's why we looked at it, because that's well known. So we wanted to see, does our mutation affect the mitochondria? And you can see these are pictures of the mitochondria. So this is in a wild type. Right here, you can see several mitochondria, and then there's much less in our mutations, right? And these are just graphs to show you that. So it definitely affects the number of mitochondria. But the other thing we found is that the mitochondria are getting more oxidative damage. So we use something called a biosensor, and it starts out green and it turns red as it gets oxidized. And what we found out is, so there's just an example here. In wild type, you can see it's mostly green with a little red, but our mutant, it's less green and more red. So that means that these mitochondria are getting damaged, right? And that we think that's probably what's causing a problem with the mitochondria. So we want to find out, how is this, how is this RNA binding protein, how is it affecting the mitochondria? So what we did, first thing is we wanted to identify, so I won't go into the details, but we want to identify what RNAs are actually binding to this protein. And we did this experiment, it's called like RNA pull down, RNA-seq. We sequenced all those RNAs. We found just 63, which was great. It's a small, we were afraid we'd find thousands, but it was just 63 RNAs. And there's a handful that were related to mitochondria, but the one that had the best kind of p-values, best statistics, was called MALS1. So MALS1 is what we call a mitoribosomal assembly factor. So ribosomes, probably many of you know, ribosomes are what's responsible for making proteins, right? But these mitoribosomes specifically, they make proteins specifically within the mitochondria, right? So it's different than the normal cytoplasmic ribosomes. They work specifically within the mitochondria. And what they do, I'll skip the details, but they're very important for creating those ribosomes within the mitochondria. And so there's too much of this ribosomal assembly. Oh no, so we know that it binds to this RNA that encodes this, but it binds to it, but the real question is, how does it affect that RNA? So we did both PCR testing and protein testing, and it's easier just to look on right here to get the point across. And our wild type, there's just a little bit of this MALS1 protein. So this is the ribosome assembly factor. There's just a little bit, but our mutant, there's a humongous amount, right? So the idea then is that this binding, this RNA binding protein binds to this RNA and prevents it from being translated. That's the idea. Now that was a very interesting finding because, as I said, when we were working on this, there was just one other paper published in any animal on this gene. And what they found is very useful for us. They found, so this is RBM, maybe you can't read it, but this is RBM26 and RBM27 right here. And this is part of this thing. It's called a nuclear RNA exosome. So this is this big structure that's what's shown here, all different proteins and complexes. And this is within the nucleus. And what this thing does is it chews up, so it destroys RNA. That's something that's very well known. So that fits perfectly with our observation that the RBM26 protein is preventing expression of this MALS1 RNA. All right, that's the idea. But next thing we want to know, so that's great that it does this, but is that really the cause of the phenotype, the axon degeneration? So we want to address that question. We did two different ways. One is we can artificially boost expression of MALS1. And when we did this, we found that it causes the same phenotype. So in wild type, we see very little axon degeneration. When we boost expression, we see much more expression. So it's actually here, the beating phenotype. So there's much more of this axon degeneration when you artificially increase MALS1. Now the next experiment, which to me is much more important, is sort of the opposite. So we make what's called a double mutant. So we have our RBM26 mutant, as I told you, in this mutation. So we mutate the RNA binding protein. And as I told you, that when you mutate that causes excessive expression of MALS1, right? So there's too much MALS1. So the question we want to ask then is, too much MALS1 causing our phenotype? Now the way we can address that question is through a double mutant. So we take our RBM26 mutant and we combine that with a mutation in MALS1. And then the phenotype goes away. And that's what you would expect if that was the cause of the phenotype. Because now in the double mutant, you cannot have too much MALS1 because there is no MALS1. I've completely removed the gene. And I'll just show you here, there's a lot of different data, but I'll focus right here. This is RBM26 mutant. There's about 25% of them have the axon degeneration. This is a double mutant, it goes down to 5%, which is the same as wild type. So we've essentially cured this disorder now by removing the MALS1 gene. So that's telling us that this excess MALS1 expression is certainly responsible for the phenotype. And this is just a quick model. So RBM26, what we think the normal function is that it's preventing expression of the MALS1 mRNA. And then so if you lose it in the mutant of RBM26, it goes away. And then we're going to have too much of the MALS1, which is going to cause too much of the MALS protein. And as I said, this disrupts those ribosomes. We think that part's hypothetical, but we do know that it disrupts mitochondria for sure. And then that's something we would expect to lead to dysfunction in the axon and degeneration. So finally, I just thank Tyler Buddell he was a doctoral student in my lab, and he worked on the calcium channel. Haley Berryman was a master's student in my lab. She did the work that's still ongoing, where we're looking at the late endosomes and the calcium channel. Tamji Chowdhury is a research scientist in my lab. He did all the work with RBM26. And that RBM26 paper was published just like in November 24. So if you're interested, it's actually an open access journal. It's called PLOS Biology. So that's something any of you could access for free on the internet if you want to learn a little bit more about it. This is my hometown, Milwaukee in Wisconsin. And I also appreciate the support of the US government for our research. We also have my own university, as well as a private foundation that has funded some of this research. And I also, again, I want to thank all of you for coming to my lecture. Again, it's great to see so many students. I hope you learned something from this. And also thank you for all the wonderful questions. So I know you already asked a lot of questions, but if there's any more, I'm happy to take them now.
I want to say thanks to Dr. Christopher for this interesting lecture. We have 10 minutes for questions. Do you have questions? More questions about the role of the calcium channels or this ribosomal protein?
Yeah.
Do you know what is the function of that ribosome protein?
Oh yeah, the function of the mitoribosome. So this is, so you're probably familiar with cytoplasmic ribosomes, right? Now the mitoribosome, what it does, it's localized specifically within mitochondria, and it only translates. So it only so the mitochondria has its own DNA, right, separate from the nucleus. And there's only, I'm not sure the exact number, only about 13 genes on there. So the mitoribosome, what it does is it translates those, specifically those genes. Are you asking the function of the assembly factor?
Okay.
Yeah, so the assembly factor, I can go back to that. Whoops, this won't. Okay. Okay, so I had skipped through this, but here it is. So here's what we think happens. So normally, so this mitoribosome, it's very different than cytoplasmic ribosomes, but it still has two subunits, a large subunit and a small subunit. And each one of these is made up of the large one is like 80 something proteins. So it's very complicated and it has to go under a construction process, and it takes a long time. So what the assembly factor does is it binds to the large subunit and it stops it from assembling too early, right? Because if it were to assemble, so you have a big subunit, a small subunit, they want to attach to each other to make the mature ribosome. But if that happens too early, it's going to be bad, right? Because the construction is not complete. So this is the MALS1 protein, and it's considered like a break. So it stops premature assembly. Now, what happens is once this, here's your mitoribosome, the large subunit, it has the MALS1 attached to it, preventing premature or early final assembly. Once the assembly, so again, this has like 80 proteins in it. Once it's, proteins and RNAs, it has, a very complicated construction process. Once it's all done and it's complete and ready, then this MALS1 will be dissociated. That's what this arrow means. It dissociates, and that's what allows the final large and small subunit to come together. So it's a break to temporarily halt binding. Now, what we think, in our mutant, there's too much of the MALS1 protein, and you can see that here. So it's bound to it, but there's also a whole bunch extra. I'm just showing 3, but I think you remember from my Western blots, it's flooded. There's a humongous amount extra. And if you think about having all that extra, it would probably prevent dissociation, right? So this is supposed to dissociate and come off. Well, if you have too much of it, that's going to be hindered, right? It's not going to happen. And that could prevent the large and small subunit from coming together. So again, this is hypothetical thinking, but to me, it makes a lot of sense. And that's our model. Yeah, thank you.
So,
Yeah.
This phenomenon has been predicted in the attention deficit.
Oh yeah, so the MALS1, no, it has not. It has not, and we have not found any, but we don't know. It may be something that's just missed. It may, in the future, it might be. But the calcium channel, though, that I started talking about, so the RNA binding protein has not been linked to, not yet anyway, attention deficit, but the calcium channel is, the first part of the talk. that's definitely been associated. In fact, it's associated with pretty much every neuropsychiatric disorder. But yeah, the RNA binding protein is maybe more specific, but I also think it just hasn't been discovered yet, that it's probably going to have a wider, I actually read a paper recently, this is after our paper was published, where there's some evidence that it might, I forgot the name of the disorder, but it's, yeah, I forgot, but it's something like, yeah, sorry, I forgot, but it's some other kind of neuropsychiatric problem, and there is some genomic evidence that it might be, it's still weak evidence, which is why I'm saying I think there's more to be discovered in the human genetics. Yeah.
We have another question in internet. Lisbeth Gonzalez Herrera asked, does any other than G35R type of mutation at the same gene of calcium channels have the same functional effect in C. elegans, given that in humans are different the novel mutations that are involved with the autism?
I'm sorry, could you repeat that once?
Yeah. Does any other type of mutation that G365R, yeah, in the same gene of the calcium channels, involved in this functional effect in C. elegans?
Yeah.
Because in humans, there are different novel mutations that are involved in this disorder.
Yeah, that's a really great question. Thank you. So here's how it works. So I told you earlier that there's these common mutations that are involved in autism. And those, we can't test them in C. elegans because they are within introns. And the introns are not conserved. So unfortunately, we cannot test them in C. elegans. That's the common variant. And we're actually proposing trying to use induced pluripotent stem cell neurons from humans to try to address that. But that's a difficult, expensive experiment, so we don't have the funding to do that yet, but it's something we're proposing. Now so that's one thing, there's these common mutations, but there actually are other rare mutations, and we have tested some of those. And actually I can show you.
Wait.
Okay. Let's see. So we've only tested one of them so far. Here it is. So this is the one, the G365R, and this one is very, very similar. It's a G365S. So it's the same residue, but it's a different mutation, and we found it had the same effect. But there actually are many other, even within Timothy syndrome, there are many other mutations, and we want to test those, but we haven't done it yet. And there's also other voltage-gated calcium channels that have been implicated in autism, and that's something else that we could test, but we haven't done it yet. But thank you.
Thanks. More questions? No? Okay. Thanks a lot for this lecture, Dr. Christopher. We have outside to the auditory a coffee, cookies, water. Yeah, can you go? Or if you want to establish a conversation with Dr. Quinn, also you can do it in this time. Thanks to everybody that's coming to the lecture.
Yeah, thank you, everyone.